I'm Mohammed Bentires Alj. People call me Momo. I'm group leader at Friedrich Miescher Institute in Basel. My lab focuses on the cellular and molecular mechanisms controlling normal and neoplastic breast stem cells, resistance to targeted therapy, and progression to metastasis. I'm pleased to welcome you to conversations with key figures in mammary gland biology and breast cancer.
Thank you. And so I'm Bob Cardiff, and I'm at this interview currently 77 years old and at the end of a really exciting career. So for the last 20 years, I've basically made a career out of being the mouse pathologist for the scientific superstars. And this has been an absolutely marvelous adventure, because here I am at a terminal point in my career, and I still have a viewpoint and a talent, a skill that is useful to the scientific community. So I have a small little niche in science, one of the few surgical pathologists. I'm an MD, so a surgical pathologist who has ever looked at a mouse. And therefore I can compare the mouse diseases to the human disease, and I can compare and contrast. So in the last 20 years, I've accumulated over 30,000 cases of mouse diseases, and primarily cancer. That's my forte, is looking at the cancers. And I've developed telepathology and digital pathology so that I can provide a service throughout the world. And fundamentally, I've got clients in 18 different countries, including Momo here, who lives, of course, in Basel, or works in Basel, and lives in France. And we communicate very well using digital pathology.
Absolutely, absolutely. Bob, so we first met at the Mammary Gland Gordon Conference in Rhode Island in 2003, where you gave a talk comparing mouse mammary tumors and human breast tumors. At that meeting, I immediately felt that the mammary gland and breast cancer community is great. Do you still remember your first mammary gland Gordon Conference, and can you tell us more about this must-go meeting?
Well, first of all, the Gordon conferences are marvelous, because, in fact, what they are is a opportunity to bring together a very diverse group of scientists who are all focused on the mammary gland, but you are meeting people with various viewpoints, and spending a week at the conference talking to each other. So it's a wonderful place to get new ideas and meet new friends.
Do you remember your first one? Or
yeah, the first one was in New Hampshire, and I think it was Colby College, and okay. Actually the first one was before I really got involved in transgenic animals, but I was still involved in breast cancer research, so okay. Yes, I do.
Bob, so you are a distinguished professor at the Department of Pathology at UC Davis, California. But before talking about your scientific career, can you tell us a bit about your personal history, birthplace, childhood?
Yeah, I went to the University of California, San Francisco Medical School. And in fact, that completed the circle, because I was delivered by a third-year medical student at UCSF. And on that particular day, my mother did not have money to put into the trolley, and she walked 21 blocks in labor to deliver me. So, yes. And I grew up in a small community south of San Francisco on the Monterey Bay called Santa Cruz, which is best known for recreation and surfing. And my father was a pest control operator in Santa Cruz and had his own business, and expected that I would go into the pest control business. But I was very intent on being a professional baseball player.
Oh.
And so I went to a school, Cal Poly in Southern California, and started a curriculum that led me to entomology. And
so we'll talk about Antonio soon about this. You know, what made you go into science and not art? Or I know you are a good basketball player. Now you said baseball. I know you are a jogger. What made you go to science instead of a sport career?
Well, I found out that I couldn't throw a 95-mile-an-hour fastball at 5'8" and 150 pounds, so I had to seek something else. And one of the things that happened to me early was that I used to go to the library a lot, the public library
in Santa Cruz.
And the librarian took a liking to me because I would come in every Wednesday and exchange books. And so she gave me a book which was the biography of a famous physician, and that biography stimulated me to think about science really the first time. And of course, going into entomology, that's a scientific career, and so I was directed towards science in one sense or another all along.
So you are an academic pathologist, but you received your bachelor's degree in entomology, right? This is the scientific study of insects.
Right.
And then you did your PhD in zoology in Berkeley in 1968. Entomology, yes, is the scientific study of insects, but what's about your bachelor's studies? What did you work on?
Well, my bachelor's studies were simply entomology, but there was an important thing in this because, in fact, I met a professor of entomology who was studying insect pathology.
Interesting.
And he was Steinhardt, Dr. Edward Steinhardt, and he started a museum of insect pathology. And what was really marvelous about him was that he was a historian, and so he told us how insects really started pathology.
Okay.
And so with that background, as soon as I got into medical school and learned about pathology, I said, whoops,
okay,
that fits.
But that's clear. Pathology, if you don't look, you won't see.
Yeah, there you go.
So what's about your PhD project? What did you do? I think it was in zoology.
Well, okay, so that's a little bit misleading because actually what happened, I went through medical school and I went through my residency, and during my residency I met a well-known mammary pathologist, Sefton Wellings, who became my mentor, and I did some research projects with him, mostly electron microscopy and immunohistochemistry. And so he recommended that I get a fellowship and do a scientific fellowship. And so I went to the usual places and interviewed, and you know, I could be a pathologist. But I went to Berkeley at Welling's urging, and I met KB Diome, who was then Medina's mentor. And Diome listened to what I had to say, and he said, well, that's very interesting, Bob. He said, you know, everybody that you've talked about said that they're going to make you a good technician, but what you really need to do is learn how to design an experiment. When he said that, I sort of perked up and I said, hey, he's right. You know, electron microscopy is a technique.
Yes.
And so, okay, I signed up with Diome and went to Berkeley. Now, what's important there is that Diome was running the Cancer Research Genetics Lab, and he was doing breast cancer research with the mice.
Yeah.
And that was the origin of my
So
background.
he's the first author on the famous paper describing the cleared fat pad.
He brought up the cleared fat pad. And of course that made Dan Medina's career and really a large part of mine.
Okay, so then you obtained your MD degree from UCSF and were trained as pathologist at the University of Oregon, Portland.
Yes.
Can you tell us about these years? Was it this time when you started really working on cancer?
Well, so I was under the influence of Sefton Wellings in Portland. So he was chair of the department up there, the pathology department. I was doing my residency, and that was his interest. And so if I'm going to work with Sefton, I was going to work on cancer, so I did.
So in 1968, you published your first paper on mammary tumors. The title was In Vitro Cultivation of the Mouse Mammary Tumor Virus, Detection of MTV Production by Radioisotope Labeling and Immune Precipitation. It was published in PNAS. What was the rationale and what were the goals of these studies back then?
Well, that's interesting because when I started my graduate work, it was said that you could not culture mouse mammary tumor cells and get virus.
Right.
You couldn't produce virus. And so at that time I knew enough about molecular biology that I thought, well, okay, let's try radio labeling. And in fact, Peter Duisberg, who has become famous and infamous, at that time had just proven that the mouse mammary tumor virus, like all of the retroviruses, had a dual or a dual genome. So anyway, so the obvious thing was to put things in culture and radio label. And so then I, in order to prove what the label was, I used immunoprecipitation and developed a radioimmunoassay.
Okay. So after your training in Oregon, you joined the University of California, Davis, I think since 1971, where you are now professor. Was this the time when you started working really on mammary tumors as an independent scientist?
Yes, I had a little interlude there because that was during the Vietnamese War, and I had the pleasure of going into the Army, and I spent 3 years at Walter Reed Hospital doing arbovirus research. And so then when I came out, actually by that time Sefton Wellings had moved to UC Davis. He invited me to join the faculty. I did, and I was there really to be their breast pathologist.
Okay. Who were maybe your role models and your mentors, and what did they do that was appealing to you?
Well, you know, I think that there's many people who have influenced me, and I think of K.B. Diome. You know, Papa Diome was one of these marvelous people that would call you into his office, chew you out for some misdeed that you had performed, a terribly designed experiment, and you'd walk out of his office and think, wow, he's really a nice guy. And so he had a marvelous way of turning even negatives into positives. So he was lovely, and he was a very clear thinker and very critical thinker. My other mentor, of course, was Sefton Wellings, because he started me going in mouse mammary tumor biology and in science. And the aspect of Sefton that I always appreciated was he had a way of encapsulating concepts that were so brief and so clear, so he'd illuminate concepts that I still have trouble explaining. So he was sort of the Thomas Wolfe of science to me. And I can think of many other people that have come along that everybody sort of gives you something.
Yes.
And so those are certainly examples. I tell you, two other people that pop into my mind is Bill Muller, who's at this particular meeting
Yeah.
with us. And the lovely thing about Bill, he's never seen a bad experiment.
Yes.
And when this experiment doesn't work the way that he thinks, wow. And all of a sudden he's off on an entirely new field of research because he grasps the significance of a presumed failure, while the rest of us say, oh, geez,
Yes.
it didn't work.
So
Bill goes, wow!
So Bill's glass is always half full.
Oh,
But we'll come to Bill in a few. So during your first 20 years at Davis, you published numerous papers on MMTV, mouse mammary tumor virus, including 2 Cell papers in collaboration with Harold Varmus and Yamamoto. Was it all about MMTV at that time?
Well, the grants that I got primarily dealt with the mouse mammary tumor virus, and then I actually went down to San Francisco and did a mini sabbatical with Harold and Mike, and which is another little story because the way things were at UC Davis at that time. I came up for merit promotion, and my department voted against me because I was doing this strange work with 2 guys that are working with chickens and something called an oncogene, and it had nothing to do with human disease.
So you proved them wrong.
Well, it took a little while.
Oh, wow.
Fortunately, cooler heads prevailed, and the department was over.
Can you tell us more about MMTV and mouse mammary tumors in the wild?
Well, so there's a couple different things. One is that very early on, I started using MTV as a marker. And so once it was clear from work of people like Harold and Mike that the virus was integrated into the genome, that we could use it as a marker. And so a lot of our work was spent looking at integration sites and following the history of cells and proving that in fact the tumors were clonal in terms of MTV. So I think that was the major emphasis during that time.
Yes. And is it found in the wild?
The wild. The answer is yes. Not universally. And in fact, the focus has really been on the wild mice that don't have MTV, because that proved that the virus was not necessary for life.
Yes.
But it was an exogenous element. Yes, you can find animals that do have the virus, but when you think about it, the virus doesn't affect their reproductive years. It's only later.
Yes.
And so it wasn't affecting evolution.
So in 1983, you wrote a review on the MUMTV, human interrelationship. Does it really matter? What would you say today about this question? Does it really matter?
This is a funny subject because every generation has a new technology that they apply to the problem of the virus and human disease.
Yes.
And so every new technology is inappropriately applied to human breast cancer, and somebody finds that there's an antigen or a sequence or something that resembles the human or resembles the mouse mammary tumor virus, but no one has ever proven that there's a relationship. And basically that article was analyzing that kind of set of data. And I must say that there's been a lot of people who ruined their careers in the search for a human cancer virus.
So what did we know back then in 1983, 85? What did we know about human breast tumors?
Relatively speaking,
Yes.
very, very little.
Compared to other tumors or in general?
No, in general.
In general.
And you know, think about where we are now with molecular signatures and
Yes.
molecular classifications of the breast cancers. At that point, there were people who were saying, well, there's only one kind of breast cancer, and those of us who practice pathology said, geez, I can't find any similarity between this
Yes.
breast cancer and the other. But that was sort of the way that people thought about them.
But then oncogenes were discovered. Do you remember when you first heard about oncogenes?
Oh, yes, absolutely. And so oncogenes and proto-oncogenes, absolutely. So I was a graduate student at Berkeley when all of that really hit the headlines.
Yes.
And the marvelous thing about all of this is when you look at all the oncogenes that have been described, where did they come from? Chickens. And rat.
Yes.
And that's about it.
Yes.
And so people ask me, well, why do you waste your time doing comparative pathology?
Yes.
Well, look what they instructed us.
So your first paper with Danny Medina and Gil Smith was in 1987. Do you remember how you met them and what was the paper about?
Well, okay, so for Dan, when I arrived at CRGL at Berkeley, Dan was already a graduate student there.
Okay.
And so he was about a year ahead of me in graduate school. And then Gil came as a graduate student from Rhode Island and gave a talk on electron microscopy of MTV. And so that was my first exposure to Gil. And somehow we sort of bonded and kept communicating all through the years.
Yes. So I know that he visits you each time he comes to, or you visit him each time you go to Washington.
Well, Gil comes out sort of yearly to San Francisco to cell biology meetings, and he's been a very good friend of a former colleague of mine, Larry Young.
Yes. And
as it has been Dan. In fact, Larry named his daughter Danielle after Dan Medina.
Wow. So, Bob, your lab played a key role in the development and characterization of the now widely used keratin 8 and 18 antibodies. Can you tell us more about this?
Well, that was another mistake. I had a graduate student who wanted to make monoclonal antibodies against breast cancer. So we started out with MCF7 cells, and he made antibodies against surface epitopes and something that was secreted. And so the good news was that he made beautiful antibodies that were very effective in immunohistochemistry, and we made radioimmunoassays. And the other piece of good news is that Ray Chan, then a fellow, eventually identified what the antigens were. And the bad news was that we identified keratins.
Oh wow! So that's how it's how you came to that.
And the amusing thing about it at the time, keratins were not supposed to be secreted, or out in the media or in the bloodstream, and they weren't supposed to be surface molecules. You know, they're intracytoplasmic. And so we had a heck of a time getting that paper published, but then we got a patent for the keratins, and the Europeans almost immediately sued us.
Oh, I didn't know about all of this. Yes.
So, you know, the good news was
the paper is cited.
You know, we got the keratins. We had good antibodies, but we were so far ahead of the wave that nobody realized that we were right.
Yes. So later, then in 1989, you described that HRAS mediates neoplastic development in mouse mammary epithelium. Was it the start of the genetically engineered mouse models?
Well, it certainly wasn't the start of genetically engineered models, but the technology had become available, and what we were interested in doing is trying to develop a organ-specific model. And of course, all the different promoters weren't available at that particular time, and so we started transfecting and infecting normal mammary epithelium with the idea that that would be a way of getting an organ-specific phenotype. And actually, Paul Edwards in the United Kingdom has really followed that up and sort of made a career out of that kind of technology. But it was an attempt to engineer the mammary gland rather than the mouse.
So in 1990, you went to Harvard Medical School in Boston, Massachusetts, as a visiting professor of genetics. Can you tell us about this experience? Was it there when you started working with Phil later?
Yeah, that was a marvelous experience, because I can recall going there with the idea that at that time, what one did is you isolated and cloned and sequenced the gene, right? So that's what I was going to do. But when I got there, I found that what they really needed was somebody who knew histopathology. Exactly. And so, yeah, I cloned, and I never got around to sequencing, but I cloned Kit.
Yes.
And that was done. But I found I was in great demand just to look at slides with people. And one of the things that happened there was that I looked at the slides that they had already collected, thousands of them, from Mick, Ras, and New.
Yes.
And I was astounded, because even though they had competent pathologists looking at them, I could see that there was a clear-cut morphological difference between the cells, the tumor cells that were being produced by these different oncogenes. And that really started me thinking and trying to expand my horizon.
So your first paper with Bill Muller was in 1991, and this was a start for a long-term and fruitful collaboration and friendship between your groups, between you and Bill. How did you meet?
Well, actually, I met him in passing when I first did a seminar at Leader Lab. Okay. And so I sort of knew who he was and so on. But the real meeting was in absentee. So I got to Leader Lab just when Bill had left, and there was a big squabble amongst his postdocs about who would get
the lucky bench.
The lucky bench.
The lucky bench.
Because Bill Muller was the luckiest of everybody. And so Phil solved it by giving it to me.
Oh, wow.
Which was resented very highly. Everybody let me know. And they had a tradition at the Leader Lab that at 4 o'clock, the latest member of the lab had to go down and get coffee and donuts for everybody.
Okay.
And they'd have tea time in the afternoon. And so, all in good fun, they assigned me the endless task of going almost the entire time I was there. I had to go down and get the tea and coffee because I was in Bill Muller's
bench.
But then what actually happened was that Muller had created a lot of these mice, and so I included in the landmark paper about genotype and phenotype. I included Bill and some others in that, and we connected and lived happily ever after.
So in 2000, I remember reading your paper in Oncogene on the mammary pathology of genetically engineered mice, the consensus report and recommendation from the Annapolis meeting. What were the major recommendations of this meeting, and are these recommendations still valid? What other recommendation would you add today?
Well, at that particular time, I think that the key focus was the fact that we were able to distinguish between the different phenotypes as related to the initiating oncogene. The other issues really involved around whether or not there was DCIS or carcinoma in situ, and we developed a definition which involved focal atypical lesions that stands today, and we also pointed out issues concerning metastases.
Yes.
And unfortunately, those issues still exist.
Yes.
And as you well know, one of the sort of defects of the mouse tumor systems is that they don't metastasize as readily as you see in human breast cancer. On the other hand, they're very explosive, they grow faster, and nobody does survival surgery.
That's correct.
So you don't know really what their ultimate would be. So
that's correct. So when studying the effect of specific genes on mammary gland development or cancer, would you advise to backcross mice to a pure background? And what's the rationale for this? Because humans are not inbred.
Well, I think there's different kinds of answers to that, and it really depends on what your scientific question is. I'll agree that complex traits kind of crossing is needed to really deal with this kind of issue. On the other hand, the real advantage of the mouse, in my mind, is you can study the biology.
Sure.
And one of the ways you can study the biology is by transplantation. And you're not going to do that if you have the mixed background.
Otherwise, you have to go to immunodeficient mice, and then you lose the effect of the immune system.
So you really can't study the biology. And that's still missing, even though everybody's in love of the patient-derived xenografts at this particular junction in history. We'll run out of that one because you can't study the entire biology of the disease.
Correct. Unless one humanized the mouse, the immune system.
Oh, well, even, yes, you can get closer and closer, but no.
Yes. So, starting with the MMTV polyoma middle T model, you generated the MIN model.
Yes.
That's correct. So, can you tell us more about the MIN model?
Well, there's a few things about the MIN model. First of all, at that time that we started developing it, that we had spent a lot of time providing services to a variety of people, and I didn't want to jump into the field and compete with people that I was already working with.
Okay.
And so I sort of surveyed the field, and what was obvious was that nobody had really recapitulated the work of Diome and Medina using the transgenic animals. And the transgenic animals gave us a very unique possibility because the initiating oncogene was genetic, it was already there, all the cells have it.
That's correct.
And so why not characterize the kinds of biology that you would get out of the precancer by establishing a transplant system where you could study it.
Yes. So there are, I think in 2000, you also compiled a CD-ROM on mammary cancer in humans and mice, a tutorial for comparative pathology. And later, I think you pioneered a website on mammary gland biology and cancer. Are these tools still available to the scientific community?
We have a website. It's available. I've run out of the CD-ROMs, unfortunately, but a major aspect of what I've done over the years is I've worked with people like Momo and others and their graduate students. Parenthetically, while I started out, I said I'm the pathologist to the superstars. You know, I don't talk to the superstars. They just send me their graduate students and fellows. Mm-hmm. And so I keep on answering the same questions. And a lot of the labs do not teach histology and pathology. And so a CD-ROM was an obvious thing to do, a tool. The website that we have is an obvious way of providing background information. Yes. And in fact, now we've put this into a class.
I will come to it. Yes.
And there's 90 hours of classwork that we've got available.
Yes, that's correct. So you are currently the director of the UCD Center for Comparative Medicine's Mutant Mouse Pathology Facility. This is the MMPF. And your research interests have focused on experimental pathology of mouse tumorigenesis, right? So you developed an archive of samples from over 29,000 mutant mice from over 200 labs from 18 countries. This is huge. This is quite impressive and extensive collection. Can you tell us more about these murine models of mammary cancer that you developed, that you collected over the years?
Okay, well, first of all, this isn't simply mammary. This includes prostate, any genetically engineered mouse that has cancer we've got collected. And we also have examples of other diseases that are built into this that we use really for comparison purposes. Okay. Probably the unique aspect of this is that the archives are ultimately all open, and they all involve whole slide imaging or digitized whole slides, which give us the opportunity to interact around the world with colleagues who use our services.
Yeah. 18 countries, yes. So Bob, you are probably one of the few surgical pathologists who combines the knowledge of human pathology and mouse tumor pathology. Do the mouse models recapitulate features of human breast cancer? I'm sure this question was asked hundreds of times.
Yes, the answer is yes. So there's a number of models that I can show a pathologist, be they veterinary pathologist or human pathologist, that are morphologically and cytologically identical. Mm-hmm. And certainly the ERB2 model, the E-cadherin P53 model, some of the MIC models are all examples of these kinds of models that unless you have peripheral clues or know what species they come from, they're spitting images of the human phenotype.
Yes. So human breast tumors are quite heterogeneous. How do you think one can deal with the complexity of this disease, and how often the mouse models recapitulate this heterogeneity?
No matter what I say, the tumors don't read my book, and so they don't know how to behave properly. And heterogeneity in cancer is an ongoing problem. Human disease is heterogeneous. We just finished publishing a paper that involved 3 cell lines from mouse, and we can show you in those tumor heterogeneity. Even though they look the same, when you look at their immunohistochemistry, they're remarkably heterogeneous, period.
So you are the founder and director of the Center for Genomic Pathology, which is devoted to education in comparative pathology. This is an online teaching tool, right? Can you tell us more about this online teaching tool? tool?
Yeah, so as a part of my interest in education, that we realized that most of the famous people in comparative pathology are retiring or have already retired, and so it was an effort, and it is an ongoing effort, to capture that wisdom, really 40 years or more of comparative pathology, and to bring in the experts, have them produce presentations, which were then developed and produced by the UC Davis Extension into really very professional and might I say award-winning presentations. And the whole idea is to allow the next generation to learn something from the greats.
Yes. So recently you attempted to focus the attention of the scientific community on the need for comparative pathologists with training in both human cancer and genetically modified mice. Can you describe to us the current situation, and did the scientific community hear your call?
The answer, yes, I can describe our failures, and no, the scientific community has not responded to the call. And it's unfortunate, particularly with people like Steve Bartels and Rod Bronson, Jerry Ward, who have really been the leaders in these fields, now facing with retirement, and you're going to lose that knowledge. Unfortunately, in veterinary medicine, the emphasis is going towards domestic animals, yes. Pet animals, and in human pathology, the emphasis is high throughput diagnosis for patient care.
Yes.
That's where the money is, and there's only a few people who are really interested in the comparative aspects of disease.
So how would you advise a junior faculty who is going to start her or his team to work on mouse models of cancer? So where are they going to find the pathologists?
That's going to be difficult. Certainly while I'm here, you're welcome to my services. On the other hand, I've got a couple of people at Davis who I'm trying to train. Whether they will stay in the field and be the next generation, we'll have to wait and see.
Yes. So, what are the major questions that you think still need to be answered in the fields of mammary gland development, mammary cancer?
You know, I think that there's two areas that we're seeing a great deal of that have not come to maturity, and just sort of briefly you can describe them as cancer initiating cell biology and epigenetics. And I just throw in that one of the major findings that comes out of Ken Diome's studies, yes. Dan Medina's studies, and our studies with the minnow model is that the transition from pre-cancer to cancer is surely epigenetic. The entire transition to malignancy is pre-encoded in the pre-cancer.
Yes. That's fascinating indeed. That's a fascinating area of research. So what would you like to tell the next generation of scientists who will watch this video?
I think that what the next generation ought to know about is that scientific discovery is one of the most fascinating things, challenging things, enjoyable things that they can possibly be involved in. And to be able to test your creativity every day is magic. And it is a absolute thrill when you find something, and somebody may have found it before, but you found it now, and it's the magic potion. Wow, is that what it's all about? And if you don't have that enthusiasm, you don't have that kind of attitude, and you find that it's really becoming a struggle, go someplace else.
Yeah, science is fun indeed.
Yes, absolutely.
The French philosopher Michel Montaigne said something like, teaching is not about filling a vase, it's about igniting fires.
Okay.
Would you comment on that?
I think that's good. So, you know, having mentors like I've had, that I realized that at very critical times in my career, my life, somebody had made what seems like a chance comment to me that hit me at the right time and absolutely influenced the direction I took. And so as an educator, that's what I'm always looking for. I know that there's students out there that if I say the right thing at the right time, it's going to light a fire. And that's what we want, light that fire.
Fantastic. Bob, is there any question that I should have asked, but I didn't?
Not in 45 minutes. No, this has been fun.
That's
Yes.
Likewise.
Thank you.
Thank you very much.